welcome to this symposium that I'm very happy to chair this morning. So just before I get to the formal introduction, my name is Michel Bouvier. I am a molecular pharmacologist and the CEO of IRIC, Institute for Research in Immunology and Cancer. And we are very happy to have this symposium this morning on G protein coupled receptor in oncology. So just a few logistic things before we get started. First of all, we encourage everybody to, if they want to maintain their camera open. We hope that this will not overload the system. If it does, we may ask people to close them again, but this is always good for speakers to see reactions from the people if we can see them on the screen. For questions, we would ask you to go and do it by the chat. And I will be translating these questions to the speakers or transmitting these questions to the speakers so that, it's a bit more smooth and we don't have to, mute, unmute for everybody to come in. But there will be at the end of the symposium, there will be network sessions with each of the speakers. So there will be breakout rooms where you'll be invited to go, and you will be explained how to go into these rooms a bit later during the symposium. And then there you will be able to, freely chat with the speakers and with the other participants. So you will have to choose which room, which breakout rooms you want to be in. But this will be possible and will allow for networking between people. You may already have noticed that this meeting will be recorded, so that we have recordings of the talks. However, the networking sessions will not be recorded. So if people want to talk about more confidential things, data that are not to be revealed publicly to a large audience, these will not be recorded, but the talks will be recorded. So you will have a little post that will appear on your screen saying that this is being recorded, and they will be asking you if you want to continue or to stop. And we encourage you to hit the continue button so that you can continue listening to the presentation. So with this, I will start the formal introduction. So again, welcome to the GPCR in Oncology in Immuno-oncology Symposium of IRIC. So for most of you, probably you are very familiar with the G protein coupled receptors. And there's increasing evidence in literature for quite some time right now that GPCRs have pluridimensional roles in cancer. And this is not too surprising given the pleiotropic roles that GPCR have in normal physiology. It's not so surprising that malignant cells would hijack these functions to help the malignant cells, the cancer cells, to survive, to proliferate autonomously, also to evade the immune system, increase their body supply, invade their surrounding tissue, disseminate to other organs. So all of these aspects of malignancy, of cancer, can be helped by some of the normal functions of GPCRs, and the cancer cells have evolved to hijack these functions. In addition to this, many GPCRs were found to be mutated in a significant fraction of human tumors. And roughly 20% of the human tumors have been reported to be associated with mutated GPCRs. And I'm taking this information and these figures from papers of one of our speakers, Silvio Gutkind, who will be speaking later today, who has made a lot of contribution to our understanding of GPCRs in oncology and has made very insightful reviews on the topic, and invite you to look at his review papers. In addition to the GPCRs, which are mutated, some G proteins, and in particular GS, GQ, G11, and G13, are frequently mutated in cancers. And in addition to these mutations, the expression of both the G protein coupled receptors and their cognate G proteins have been found to be dysregulated in many cancers. As you all know, GPCRs are the target for a very large number of drugs. It's generally accepted that roughly 30% of FDA-approved drugs target GPCRs for their therapeutic function. However, there's very few that are used in anti-cancer therapy until now. As of last year, there was essentially 8 drugs that target GPCRs in treatment of cancer. And this includes even one, which is a Reloxifene, where yes, of course, it can bind to GPR30, but most likely lots of its activity goes through the estrogen receptor, the nuclear receptors, and not the GPCR, although this is a matter of active research. So there's very small number of GPCR which have been targeted for anti-cancer drugs. Knowing how they are good drug targets in general and druggable, that of course leaves a world of opportunities for the development of GPCR targeting drugs in oncology. And this is essentially the topic of today's presentation. And we are very lucky to have a list of speakers which have made very important contributions to this field. First, we'll hear from Martine Smit from the Free University of Amsterdam. And Martine has made groundbreaking work on the ability of some viruses, for example, to hijack some GPCRs, promote mutations in these GPCRs that become oncogenics. And also more recently has made very interesting discoveries about how nanobodies, a form of antibodies, could be used to modulate GPCR activity in the context of oncology. We'll then hear from Xavier Leroy from Domain Therapeutics, who is leading the effort of Domain Therapeutics in the use of monoclonal antibodies in targeting GPCRs, in particular in the realm of immuno-oncology approaches to treat cancers. We all know how immuno-oncology has transformed the treatment of cancer patients and is still underused. We all know that the potential of immuno-oncology is greater than what has been came to fruition until now. And clearly GPCRs could be an important players in increasing the ability to use immuno-oncology, and Xavier will tell us about their efforts in that direction, more generally what could be envisaged for the future. Then we will hear from Sébastien Carreneau from IRIC at the University of Montréal, who is a cellular biologist who has been doing important work on a family of protein, the ERM proteins, and will tell us about some of the work that he's been carrying out, in particular with our group, on the relationship between some G protein coupled receptors, the ERM system, and the metastasis process of cancer cells, which may be opening some avenues for development of new treatment involving GPCRs in cancers. And finally, we will hear from Silvio Gutkind, who really was one of the first person who insisted on the potential role of GPCRs in cancer. And as I mentioned in one of the slides that I showed during my small introduction, Silvio has really written important review papers on the topic. And not only review papers, but really getting into the databases and getting analysis of the mutations which are present in patients with cancer, mutation in G proteins and also in GPCRs. And so it really is, he is one of the leaders in this field, and we're very happy to have him this morning to give his presentation on the mutational landscape of G proteins and GPCRs in cancer. So Martine Smit is from Amsterdam Institute for Molecular and Life Science. And so she will be telling us today about nanobodies modulating oncogenic signaling networks activated by human and viral chemokine receptors. So Martine, I will stop sharing my screen and you can start your own sharing, and we'll all listening to your presentation.
Okay, so Michel, thank you very much for the introduction, the kind introduction, but also the invitation to share some of the work we've been doing on human and viral chemokine receptors. On this very exciting topic on GPCRs in oncology and immuno-oncology. Like Michel already alluded to, so GPCRs are truly connected to cancer. And especially the work done by Silvio Gutkind. He did some very elaborate studies showing indeed that different type of cancers express, highly express GPCRs and also mutant GPCRs. So I will share some of the work we've been doing on human and viral chemokine receptors. And we've been using nanobodies actually to modulate those receptors and to get insight into the role of those receptors in cancer. So if we look at the GPCRs, which are highly overexpressed in cancer, those are actually the chemokine receptors. And if we look at chemokine receptors themselves, we know that the chemokine receptors play a role, and they're expressed primarily on immune cells, and they are actually trafficking to sites where high levels of chemokines are released. And often at sites of inflammation, immune cells expressing those chemokine receptors will migrate to those sites of inflammation and take care of the inflammation. Yet a lot of diseases are associated with chronic inflammation, and the HIV has taken advantage of the chemokine receptors because CCR5 and CCR4 act as co-receptors for HIV. But if we look more specifically to the cancer setting, and we look to the different types of cancer types out there, we actually see that a lot of those chemokine receptors are highly overexpressed in those settings. And if we look more closely, it's apparent that CCR4 is expressed in almost all tumor types. And if we look more specifically at a primary tumor, the cell will grow and will resulting in an hypoxic core. And within this hypoxic core, HIF-1 alpha is being activated. And HIF-1 alpha is a transcription factor regulating the expression of CXCL12 on one hand and CXCR4. And those are the pair which is actually inducing proliferation. Well, in time, also blood vessel formation will take place, so angiogenesis, and tumor cells can actually extravagate through the bloodstream to distant sites and start a metastatic niche elsewhere. So the chemokine CXCR4 and CXCL12 axis is believed to play a role in metastasis. But not only in the tumor site itself, other cytokines and chemokines are being released and resulting in actually infiltration of immune cells to the site of inflammation, creating a so-called tumor microenvironment. And we know nowadays that this tumor microenvironment is quite essential because it's maybe pro-tumorigenic, but it may also be immune suppressive, meaning that it ensures that the immune cells cannot take care of the tumor cells. So this immune suppressive area. And in fact, two chemokine receptors play a role in this setting. CXCR4 on one hand, which is actually binding CXCL12. It's a classical GI-coupled receptor. It's highly expressed in different tumor types and plays a role in metastasis. But besides that, also ACKR3, formerly known as CXCR7, binds CXCL12 and is also shown to play a role in cancer. Especially the aggressive tumor types show also expression of ACKR3. Well, this is an intriguing receptor because it's an atypical chemokine receptor. ACKR3 stands for this, and it binds CXCL12. So it's believed to actually scavenge CXCL12 away and indirectly affect CXCR4 function. So a lot is still unknown about those two chemokine receptors, but within OncoNet, which is an European training network, we are studying those two receptors. We're trying to find modulators on one hand, try to identify the signaling networks activated by those receptors, and try to see how they play a role in the primary tumor, in the metastatic niche, as well as the tumor microenvironment. And we were lucky that we got funding for OncoNet 2. So we will keep on continue working and focusing on those two receptors. So how do we actually target chemokine receptors? Well, like GPCRs, we can target them with fragments growing into small molecules, targeting the receptors of interest. We can make use of peptides, but we can also make use of biologics. And in fact, the biologics have become increasingly important in drug discovery, and also biologic targeting GPCRs. So, and with the biologic, we mean the monoclonal antibodies, but also the antibody fragments, the so-called nanobodies. And so what are nanobodies? So they are small antibodies.
Martine, can I interrupt? You're still on the first slide, so I don't know if you have moved slides or not, but we're still seeing only your first slide.
Oh, really? Oh my God. Yeah. Okay, good. So I was focusing on the primary tumor. So CXCR4 is highly expressed in this setting, serving as a metastatic niche, and the other receptors ACKR3, and both play a key role in our OncoNet consortium. So if we look at the GPCR ways of targeting, so small molecules, peptides, or biologicals. And so the biologicals have increased insight into the targeting of GPCRs, and especially the use of nanobodies. So with respect to nanobodies, so nanobodies are actually the smallest part of the so-called conventional antibodies seen over here. And especially the camelid family has expresses besides the conventional one, the heavy chain only one. And the nanobody is the smallest part. It's a fragment, can be obtained through immunization or through use of a synthetic library. It's highly selective and highly stable. And the nice thing about a nanobody, it's small in size, is a single domain. It can be efficiently selected for, it can be selectively be produced. And the nice thing in a cancer setting that nanobodies do integrate penetrate into the tissue and into the tumor. And the key findings of nanobodies are that they have flexible and long CDR regions and binding into cavities. So they actually recognize conformational epitopes. So if we look in this slide, there are different nanobody GPCR applications. So for the intracellular binders, those are nanobodies targeting the GPCRs of interest. They act as so-called stabilizers, and they've been instrumental in the finding of X-ray structures done studies by Kobilka and Jan Steyaert. But the intracellular binders serve also as biosensors, and they have been instrumental in looking at GPCRs, especially signaling from endosomes. But besides the intracellular binders, we also see the extracellular binders, and they serve as modulators. So they bind extracellularly, and they can actually inhibit or activate GPCRs. Moreover, those nanobodies can actually be extended with the FC chain to engage the immune system. They can be coupled to fluorophores, and they can be used for imaging. And a more elaborate presentation on the use of nanobodies in GPCR setting can be actually viewed on the webinar of Domain Therapeutics on their website. So looking at CXCR4, as I showed you earlier, it's highly expressed in tumor setting. So together with Eblings, we've first identified nanobodies targeting the CXCR4 receptor. And so we looked for CXCL12 displacers. So here D2 and D4 are two nanobodies which do bind the receptor and displace CXCL12 from binding to its receptor. And so we know the epitope of these nanobodies. They bind to extracellular loops. And the nice thing is we link them, creating bispecific nanobodies. And those bispecific nanobodies have the advantage that they show high affinity for the receptor, and they show high potency. And as can be seen here, they inhibit CXCL12-induced migration. So the bivalent, bispecific one have increased potencies. But also on a constitutively active CXCR4 receptor, the monovalent does not change the signaling behavior of this receptor, while the bispecific one does so, and it inhibits the basal activity. And very nicely in a monkey model system, those bivalent nanobodies do induce stem cell mobilization. So, and they appeared as effective as AMD 3100. But the nice thing about those nanobodies as well is that you can reinsert immune functions. So what we did, we have two nanobodies here, and we added a FC chain to it. So in this sense, we included FC effector functions. So we could actually induce antibody-dependent cytotoxicity on one hand. And if we look at CXCL12 binding, the monovalent has affinity for the receptor, but the FC one has a much increased affinity for its receptor because it has a higher affinity. So that has an advantage. And the FC chain allows it to bind and recognize FC receptors on, for example, NK cells, so which are natural killer cells. And once those are activated with the FC chain, they will release compounds which will kill the cancer cells types, and the cells go into lysis. And especially we're studying this right now within a consortium, magnetic, in which we study the role of CXCR4 in leukemia. And in leukemic setting, a CXCR4 expression normally in B and T cells is low. However, in a leukemic setting, we see an overexpression of CXCR4. So they are highly enriched with CXCR4. And when we incubate those cells with the FUN 400, which is the CXCR4 nanobody with the FC chain, what we see is that CHO cells, which do not express CXCR4, are not killed off in the presence of NK cells. HEK cells showing low expressions do not show any killing of cells. Yet the tumor cells, which highly express CXCR4, they will bind the CXCR4 nanobodies with the FC, and in the presence of NK cells, those cells are killed off. So showing that we can actually reinsert the immune effector functions. Next to that, we also identified nanobodies targeting the ACKR3 or CXCR7 receptor. As said, it's an atypical receptor binding CXCL12 or scavenging CXCL12 and recruiting beta-arrestins. So we identified nanobodies targeting this receptor. So they would bind the receptor and displace CXCL12, and we identified several ones. And those which were binding to the ECL loops, in nanobody 2 and 3, were able to inhibit CXCL12-induced beta-arrestin recruitment, whereas the N-terminal binders do not. And very nicely, in an in vivo setting in which we used head and neck cancer cells, which highly express ACKR3, when we inject those cells into nude mice in a xenograft model system, those mice develop tumors in time. Mice which were actually treated with those trivalent nanobodies which have an extended half-life, they showed impaired tumor formation. So showing that by targeting ACKR3, we can reduce tumor formation. And when viewing and looking at the tumor setting thereafter, and we looked for angiogenesis, so blood vessel formation, we saw that the mice which were treated with the nanobodies targeting ACKR3 showed reduced angiogenesis, so blood vessel formation. So apparently by targeting CXCR7 or ACKR3, we can reduce angiogenesis. So that those were examples targeting the human chemokine receptors CXCR4 and 7. But what we also know that in fact, viruses have hijacked the chemokine receptor system. And in particularly the herpes viruses. So herpes viruses are DNA viruses, and they contain one or more genes encoding for genes targeting GPCRs, encoding GPCRs. And so examples are the EBV virus, cytomegalovirus, CMV, and Kaposi sarcoma herpes viruses. And they all contain one or more genes encoding for GPCRs. So our lab is interested in CMV virus, encoding 4 different GPCRs, and more specifically, they look and resemble to chemokine receptors. And so what are those hallmarks of those viral GPCRs? Well, they are expressed already on the virus and after infection. They bind a broad spectrum of chemokines, and most notably, they display constitutive activity, so basal signaling. So without a binding of a chemokine, they can already signal. And moreover, they show promiscuous G protein coupling. While the classical chemokine receptors couple to GI, the viral GPCRs couple to GQ mostly, but also GSI1213. So they have the whole spectrum and activating downstream signaling. So at first, they were believed to actually play a role in immune evasion because they were scavenging the chemokine away from the environment. However, now we know that they play a role in inflammation and cancer as well. So what's the link between CMV and US28? So looking at sites CMV infection, so 50 to 80% of the population is CMV positive. Mostly, this CMV virus is present in a latent format and asymptomatic, luckily. However, when you are immune compromised or undergoing organ transplantation, then CMV may be reactivated and have serious side effects. So the CMV pool is primarily present in the CD34 and CD14 positive monocytes in a latent reservoir. And in time, different labs in the US and in Sweden had discovered that CMV proteins were apparent in different types of cancers, glioblastoma, breast cancer, colon, and prostate cancer. And we were actually intrigued by it. So why does such a virus express those viral chemokine receptors? And we focus at first at US28. It's constitutively active, it's showing ligand-induced signaling, it's expressed in glioblastoma, and it plays a role in latency. So as said, it shows homology to the chemokine receptors, especially CCR1 and fractalkine receptor. It binds a lot of chemokines, so acting as a chemokine sink. And as such, it was actually postulated to play a role in an escape strategy. It's a functional GPCR showing constitutive and ligand-dependent signaling, and is expressed under latent conditions. And the Chris Garcia group co-crystallized fractalkine with the US28 receptor. So that information is available right now as well. So looking at the activity back quite some years, my first PhD student, Paula Casarosa, she had shown that by increasing the concentration of US28, she showed increased signaling on inositol phosphate production, so GQ readout, as well as NF-kappa B, which was not the case for the classical chemokine receptors. But also in a CMV setting. So we infected glioblastoma cells with the virus, a wild-type virus, which is intact, and a delta US28 virus, which is not harboring this US28 gene. And if we look at GQ activity in infected cells, we see that those infected with a CMV virus show this increased inositol phosphate production, which was not apparent for those not expressing US28, clearly showing that an infected setting is showing this increased signaling. So we went on studying this receptor, and it was clear that fibroblasts expressing US28 were highly proliferative, whereas the uncoupled G protein in the famous DRY motif was not showing this signaling. It's oncogenic, so showing transformation, so foci were being formed, and when we inject those cells into nude mice, tumors were formed. We looked at the microarray analysis, and we looked at which genes were highly changed and upregulated, and especially the NF-kappa B related genes driving COX-2 and VEGF were highly upregulated and highly secreted. VEGF was secreted. And together with the school, Sergio Lira at the Mount Sinai School of Medicine in New York, mice expressing US28 in the intestine showed neoplasia in the intestine in time. But most importantly, together with Cecilia Söderberg-Naukliker of the Karolinska Institute, we observed that US28 is in fact present in brain tumors, glioblastoma, and especially in the vascularized areas. So that was actually kind of a really breakthrough that really in patient materials, we could detect viral chemokine receptors. So if we look at glioblastoma, it's a very aggressive brain tumor. Survival only up to 15 months, and the current therapy is surgical resection, radiotherapy, and chemotherapy, increasing the survival of about 12 months. But Cecilia Söderberg-Naukliker had already detected the CMV in the glioblastoma and brain tumors. And so she started a small clinical trial in which she treated those glioblastoma patients which were CMV positive with valganciclovir, which is an anti-CMV agent. It blocks viral DNA synthesis. And along with the radiotherapy and chemotherapy, she saw indeed that when they were on valganciclovir, there was an increased survival of at least 2 years. And now she has expanded the cohort and trying to link really the CMV and onset of cancer. So we got interested. So why do those viral GPCRs, do they play a role? And if so, do they indeed increase this cancer progression? So we looked more carefully at glioblastoma. And one should note that the CMV protein expression is only present in the tumor area and not in the healthy surroundings. So when we infected glioblastoma cells with the CMV virus, we saw that the 3D growth was really extensive, which was not a case when they were infected with the virus without US28. So no increase was observed. Moreover, US28 itself can do the job already. And if we look at what is being secreted, VEGF was highly secreted. And when we looked into our intracranial model system, in which we injected those tumor cells into the brain of mice, we see normally that the U251 cells not expressing US28, after 60 days, they start to show tumors. Yet those expressing US28, already after 15 days, we see tumor onset, clearly showing that in an in vivo setting, tumor formation is taking place. So with our nanobody platform in place, we identified nanobodies targeting this viral chemokine receptor US28. So US28 binds CCL5 or RANTES. So we were looking for nanobodies which could displace rantes from the receptor. So here we have a monovalent which nicely displaces CCL5. We did the same trick. So we linked two nanobodies, creating a bivalent nanobodies with higher affinity on one hand. And we looked at the basal sickling of US28. So basal sickling is highly increased. GQ sickling is what we're looking at. CCL5 can further increase it, and our monovalent nanobody can inhibit it. But nicely, the bivalent nanobodies do show basal decrease of basal sickling. So they act as inverse agonists. And when we looked at 3D growth and we incubated those spheres with US28 nanobodies, we could really see that the 3D growth was impaired. And also VEGF secreted was impaired. So next we went to our tumor setting, our intracranial model system, and we treated those mice with US28 nanobodies in a trivalent format, injected them IP. And what we saw, in fact, so normally after 20 days, we already see massive tumor formation. And those mice which were treated with the US28 nanobodies showed reduced, impaired tumor formation, clearly showing that if we can target this US28 receptor effectively. However, as you can see, it's not fully inhibited yet. So we took another approach. And so we used the nanobody and linked it to a so-called photosensitizer to be used in photoimmunotherapy. So here we have the nanobody. So we linked it directly to a photosensitizer, so an RI dye, which normally is not toxic. However, when we irradiate it with near infrared, the photosensitizer will form oxidative radicals and kill off those cells in close proximity. So only those cells which will bind the nanobody will be killed off. And that's what we looked at. So we have on one hand US28 positive cells and negative cells, and we exposed them to the nanobody with the photosensitizer, irradiated them, and we saw that only the US28 expressing cells were killed off. And also here on an immunofluorescence, we see that the US28 positive cells here in blue binding the nanobody were killed off as shown by propydium iodide. And also in a 3D setting, we looked at spheroids, exposed them to the nanobodies with the photosensitizer, irradiated them with near infrared, and we checked 24 hours later to see whether the cells were still viable. And as you can see here, the US28 negative cells were not killed off, whereas the US28 positive cells showed clearly decrease in cell viability and were killed off at higher dose. And then some of our really truly novel data, which were accepted yesterday. So we were very happy about that. So we had also a program looking at so-called intrabodies. And remember, intrabodies are nanobodies which bind the intracellular side of GPCRs. So earlier on, the Chris Garcia lab with Hidde Ploeg already had found nanobody 7, which is an intrabody binding to the US28 only when fractalkine is bound. So this is recognizing the fractalkine bound receptor. We found nanobodies, FUN103, which is binding and recognizing the basal, the ligand-free receptor. And so when we actually link those to either nanobody 7 or FUN103, and we looked at binding affinity of rantes, we see that without any, in the presence of nanobody 7, we see a shift in potency, for fractalkine, so showing higher affinity for the receptor. And when we had in the presence of FUN103, active binding just the constitutively active form, we see a lowering of chemokine bound binding. So we have now one receptor which only with a nanobody 7 recognizes the fractalkine bound version, and we have a nanobody which only recognizes the ligand-free conformation. So we wanted to know, can FUN103 indeed inhibit US28 function by inhibiting GQ signaling? So what we did, we had US28, which was coupled to an enzyme, BioID, which biotinylates all proteins in close proximity. So it will biotinylate the receptor, but also other proteins in close proximity. Well, US28 is known to bind GQ, so we looked specifically at GQ, and GQ is highly biotinylated in the presence But in the presence of FUN103, the intrabody, we see a lowering of GQ biotinylation because FUN103 displaces GQ from US28. So we see a lowering, which was not apparent with an irrelevant nanobody. We looked also at mini G alpha Q recruitment to the receptor. So normally the GQ and US28 are in close proximity. We see breadth transfer. However, in the presence of FUN103, we do not see this. So because FUN103 binds to the site of the GQ protein. And if we look at GQ function, we see that in the presence of an irrelevant nanobody, we see still intact GQ sickling. However, in the presence of FUN103, we do not see any GQ activity anymore. So FUN103 apparently can really inhibit GQ function and US28 function. So we wanted to know, can we use FUN103 to bind and to reduce spheroid growth? And indeed, cells expressing FUN103 show this reduction of tumor formation, of 3D spheroid growth very effectively. And of course, FUN103 is an intrabody, so you need to make sure that your gene of interest is expressed intracellular, but with the current delivery modes, it is a way forward. And then next, another very exciting story is that of the role of US28 in latency. So as I told you, a lot of people are in fact CMV positive, but CMV is actually residing in latent pool. And US28 is implicated in this latency. So what does US28 do in this setting? It inhibits expression of immediate early. And immediate early is the gene which is important for viral replication. So what it does, US28 shuts off the viral replication of the DNA virus. And in that sense, there's no way for the T cell to recognize an infected CMV infected cell. So what we were wondering, so especially people undergoing transplants, this latency can be reactivated, and you get reactivation of the virus, which is a true problem in transplantation. So because this way, the organ can be actually rejected. So people are under immune suppressives and also on antivirals. So we wondered whether we could use our nanobodies, FUN100 bivalent, to sort of partially reactivate the virus. So the virus could actually, the T cells could redetect this latent pool. So we were asking ourselves, can FUN100 bivalent partially reactivate the virus? And if so, can it initiate clearance of this latently infected cells by T cells? And also these studies were just completed, and last week we got also the good news that it was accepted. So what we did in those studies, we had CMV positive donors in which we collected the CD14 positive monocytes. That's the pool where CMV is present. We super infected those cells with the CMV virus with an YFP immediate early tag, so we can actually easily monitor it. And we waited until latency was occurring in vitro, and we exposed them to FUN100 bivalent nanobodies in the hope to reactivate in part the immediate early. And if we look here on the right side, what we did indeed see is that cells which were exposed to irrelevant nanobodies, the immediate early positive nuclei were the same, yet looking at the FUN100 bivalent treated cells, we saw an increase of immediate early, meaning that the break of US28 was actually released. So we saw expression of immediate early. So now the immune system can detect those cells. So that's what we wanted to check, and that's what we did. So we exposed the cells to T cells, so the CD4 and CD8 positive T cells on one hand, and on the other hand, we had T cells, the PBMCs depleted of the T cells, and we checked whether those T cells could actually clear this subset of infected cells. And those cells which were treated with the irrelevant nanobody, no changes were apparent, obviously, but the cells which were actually exposed to FUN100 bivalent moiety, when they were exposed to the depleted PBMC, so no T cells, of course, the immediate early positive T cells were still same amounts. Yet those which were exposed to CD4 and CD8 positive T cells, they could actually clear the immediate early cells. So now these T cells could recognize the reactivated subset of those cells. So they could really clear this latent pool. So it's very nice to show that this anti-US28 nanobodies can trigger, on one hand, the clearing of latently infected cells. And we're now evaluating whether we could use this as a potential therapy for pre-treatment of people and patients undergoing organ transplants. And the last part, just a few slides left. That's exciting work on the convergence of exosomes and GPCR biology. Well, as GPCR people, we really know what after activation, you get internalization into immediate early endosomes. But what's also apparent is that they actually, in the late endosomes, they get incorporated into so-called multivesicular bodies, MVBs. And within these MVBs, we see those exosomes. And cells do secrete those exosomes. And the interesting thing is that we've observed that, in fact, those exosomes are highly enriched of GPCRs, and especially those GPCRs which are mainly intracellular, such as US28, but also ACKR3. So looking at U251 cells with the H-ATAC US28, so it's highly enriched. This is one cell. US28 is highly enriched in the endosomes. And if we look and use electron microscopy studies, EM studies, we see here this MVB moieties as seen over here. And if we look carefully, using gold particles against the HA tag, we see that US28 is in those MVBs. But more importantly, what about the secretion of these exosomes? And I hope I can actually activate it. Now I have to take off my laser, I guess. Oh, okay. Now it's okay. Now, okay, it's not so. We use the fluorine US28. So we tagged US28 with a pH-sensitive YFP moiety. And if we look under a microscope using high-end turf microscopy, and we look in time, we see that those exosomes are nicely released. And unfortunately, I cannot activate it now. Let me see if I can take away the laser. Yeah, now it's working. So if we look in time, we see that spots appear, and those are the pH sensitive US28 on exosomes. And in time, we see nicely that exosomes are being released by US28 expressing cells. And of course, this is a transfected cell. What about an infected cell? So what we did, we had the CMV strain, which we had the back clone. So we inserted the US28 fluorine into it. So we have a CMV, which is US28 fluorine and pH sensitive. Also in EM studies, we can still detect US28 in the MVBs. And if we look in time, and I don't have this in a mode, but if you see in time, 30 to 50 seconds, we see really release of exosomes of CMV infected cells. So apparently those exosomes are indeed released of CMV infected cells. But what is the functional consequence, and what's the role of exosomes? So actually we do not truly know. We believe that they play maybe a role in the tumor microenvironment. And so if you think and look here at the exosomes, so exosomes carry those US28 or GPCRs, and they can bind chemokines, and as such they can scavenge chemokines. So maybe this is also a way to evade the immune system. Or EVs may be taken up or can be fused and induce a cellular response. So we're still looking further into these studies, but what we can tell you is that we know that the US28 expressing exosomes, they do bind fractalkine, as seen here using Brett studies, which can be inhibited by the nanobody. And if we look at the effect of fractalkine for its receptor, the fractalkine receptor, control EVs do not affect it, but if we expose this fractalkine receptor activation assay to the US28 expressing cells, we see reduction of signaling. So apparently US28 can bind fractalkine and scavenge it away so it can no longer signal. So we believe that this US28 exosomes may be playing a role in the immune suppressive area. So altogether, I hope I've showed you that indeed chemokine receptors play a role in immuno-oncology. So I showed you examples that CXCR4, ACKR3, and viral GPCRs are highly expressed in cancer. They do play a role in proliferation, invasiveness, inflammation, angiogenesis, and they are highly enriched in exosomes and maybe play a role in tumor microenvironment. They play a role by activating oncogenic signaling networks. Well, I did not have time to go into details of that, but I can assure you they activate HIF1 alpha axis, NF-kappa B, COX-2, the IL-6, STAT3 axis, beta-catenin, and PKM2 pathway. So they clearly target the cancer hallmark pathways. And nicely, with the nanobodies in place, we can use them as modulators, as biosensors, and include FC or use them as tracers. We've shown you that the nanobodies act as important tools. It can be used for imaging, but more importantly, as modulators. So the bivalent nanobodies, they inhibit ligand-induced and constitutive signaling. They can reduce tumor growth. They can reactivate and clear latently infected cells. And with the incorporation of the FC chain, we can also induce immuno effector functions. So use them for immunopharmacology. So altogether, I think we believe that those viral and human chemokines play a role in onset of cancer, and those nanobodies can be used as tools and eventually maybe as therapeutics. But most important slide actually is actually the nanobody GPCR team who has been doing all the work, the hard work, I should say. So Sven Jenigen and David Mosson in the earlier studies together with Ebling's team. And then the Timo de Groof and Raymond Heukers did a lot about the US28 nanobodies together with Nick Bergkamp, together with Liz Elder and John Sinclair at Cambridge, looking at latency, the phosphodynamic therapy with Sabrina Oliveira, then the people at the medical center on the glioblastoma expert, the exosome expert Michiel Pechtel, together with Maarten Babelman, then the people at Cecilia Söderberg Nuclear in Sweden. And I did not have time to actually discuss any work with Michel Bouvier or Bianca Plouffe, but there we're also looking at GPCRs more closely. And most importantly, the OncoNet consortium, because within the OncoNet consortium, we're looking at CXCR4 and ACKR3 receptors. And we have a really bright team of supervisors, but also PhD students working on those two receptors. And thank you for your attention.
Thank you very much, Martine. Very interesting talk. So I will be handling the questions during the session. We already had a number of questions, so if we don't have time to go through all of them, remind you there will be networking at the end, so people can join the breakout rooms and ask their questions. But I will start with the one that were asked during the talk, and then I will continue as we go on. So the first question was from Sally Johnson, was about the bivalent nanobodies, and she was wondering if they could affect the dimerization of the chemokine receptors and their signaling activity.
For sure. So we believe in the GPCRs are also functional as dimers, and definitely the bivalent nanobodies do target both a dimer, because we have bivalent moiety, so they cannot bind the same receptor, and we believe that they really bridge dimers and higher order nanobodies. Yes. Yeah.
Thank you. So Xavier Leroy was wondering if the anti-6R7 nanobodies that you've used or antibody were targeting, tumor cells, endothelial cells, or immune cells.
That's a good question. We tried to look at it in our tumor setting, and so it's difficult to see. So on one hand, they inhibit the tumor cells themselves, but we have also the feeling that in this vascularization process, this angiogenesis, so we feel that they might also target that part as well. So it has a dual mode of action. Yeah.
Okay, so as Alicia was wondering, that she was saying that she showed great work on antibodies and intrabodies, but whether or not you thought that small molecules could also be interesting for these receptors.
So for the viral chemokine receptors.
Well, I think
Or,
both the chemokine and the, I think that at the time it came, you had not started the viral yet.
yeah, no, of course. So I'm not saying that only biologicals and nanobodies will do the job. I think still small molecules are very interesting. They're easier to provide as well. But I think the nanobodies can be of help of finding selective small molecules, because now we have a receptor with the nanobodies, which you can use as an interface to look at small molecules targeting that phase, or even at the GPCR G protein interface. So I think the intrabodies are very valuable as well, because now we can suddenly target this GPCR G protein interface very selectively. So yes, I think this opens avenues to find small molecules targeting at different interaction sites.
Okay. And then Joe Mancini was interested by the COX inhibition, in colon cancer patients, and wondering if this was looked at in terms of the response of the patients that have HCMV infections in colon cancer, and whether or not this could have an impact for the
Yeah.
responsiveness of to COX inhibitors.
Yeah. So I think since CMV indeed induces this inflammatory process via NF-kappa B, and COX-2 is highly elevated. So I think the beneficial effects of COX-2 inhibitors may in part be due to that aspect. So yes, I believe that they may be ways to inhibit inflammation. And since the virus uses the inflammatory sites, I would say yes. Yeah.
Okay. Then Emel Balagu asked, about the HIF signaling pathway. You mentioned that these receptors do indeed activate the HIF pathway, but the question was more, could you modulate it with the nanobodies?
Yes, we can, because so for US28, for example, it's highly constitutively active, and in activating the HIF1 reporter as well. And indeed, in the presence of the US28 nanobodies, the bivalent, we can see this inhibition, not the monovalent, but the bivalent, yes. Yeah.
Okay. Brian Airy is asking whether you have tried to look at the combination between the nanobodies and the checkpoint inhibitors to see if you could potentiate the response.
Yeah, not yet. So that's in the plan, of course, because I'm pretty sure targeting just the tumor is not sufficient. So you really need to target the immune cells as well. So the combination is a really, truly good one. Yeah.
Okay, and I think I'll take a last one in the interest of time. No, we still have a little bit of time, so I'll take probably two and maybe take the privilege of the chair to ask one myself. So Yuri Saragovi is asking, does the CMV GPCR intrabody alter exosome numbers, quality, function? What is the impact of the intrabody on the exosome exit in the CMV?
Um, we're just about studying that. I mean, so we now two fields of research line are converging as well. So we had on one hand our intrabodies, and on the other hand the exosomes. So now we'll see whether the intrabodies can be used to actually also target the exosomes as well. But the exosomes, the extracellular site is the outer site. So we believe that actually the extracellular binder are more effective in that sense.
Okay.
Yeah.
Alexander Hauser is asking, first of all, he congratulated, many people have congratulated you for a fantastic talk, but I just keep that because, in the interest of time. But so what about what is your thought about the stability of the nanobodies versus the full length antibodies in terms of going forward with therapeutics?
Correct. So nanobodies are rapidly cleared. Within 2 hours, they are cleared for certain applications, such as photodynamic therapy, that might be valuable. You just want to hit the tumor very locally and short time period. But if you want to really have a therapeutic means, you need to extend its half-life. And we can do so by making bivalent or trivalent. That's a nanobody against albumin. And in this way, you can actually extend its half-life to 2 to 3 weeks. And another way is to include this FC chain. And then, of course, then you are almost like a conventional antibody, and those will also reside longer. and have a longer
It's
life.
not. It's nice because you just answered the next question from Patricia McDonald, who was asking exactly if the FC could increase, FC fusion could increase the half-life. My question was when you talked about in the head and neck cancer and you use these nanobodies, the nanobodies you showed us that had an impact on the size of the tumor, they were labeled NB4 and NB5. So I was wondering if you compared those nanobodies that did not interfere with beta-arrestin recruitment versus those that did, like NB1 versus NB3, for example, and did you see a difference in terms of the response?
Yeah, that's a good thing. So so I believe that you need for CXCR7 to inhibit the beta-arrestin part anyway. So those which were most effective and before was actually targeting the ECL loops. And the ECL loops are the one which block the beta-arrestin mediated pathway, whereas the N-terminus does not affect beta-arrestin signal. So you really need to block the ECL loops.
Okay.
So
the reason why I'm asking is that there are increasing evidence that the beta-arrestin, although it is recruited by CXCR7, may not be required, for example, for the endocytosis of the receptor itself, which is sort of counterintuitive. But so I was wondering if there could be some other pathways that could also be just by blocking the ability of the chemokine to bind to the ACKR3 and then have the same function. But just wondering.
No, yeah, I definitely. So we have within OncoNet 2, we showed in that ACKR3 can internalize without beta-arrestin, so it does not need it at all. And we have been very much biased towards beta-arrestin, and I truly believe that there are other interactors which do play a role, and we should look into those as well. Yeah.
Well, thank you very, very much, Martine, for a very interesting talk. So you may have noticed that I waited until these announcements were made before starting speaking. It makes it a bit easier. Thank you, Tony. So the next talk will be by Xavier Leroy from the domain Therapeutics. And the title of his talk will be Monoclonal Antibodies Targeting GPCRs in Immuno-Oncology. So Xavier, please take it away.
Thanks, Michel. Can you see my screen? Yeah.
Yes.
Yeah.
But yeah.
Okay. Good morning or good afternoon to everyone. Pleasure to be with you today and with Michel and colleagues to speak about GPCRs in immuno-oncology. From the presentation from Martine on the discovery application of nanobodies to understand the complexity of GPCR signaling pathways and applications to the treatment of several types of cancers. Through my presentation, I will present to you several approaches that we do follow at Domain Therapeutics or that are followed by different biotech and pharmas to discover antibodies for therapeutic applications, especially on GPCRs and for immuno-oncology applications. So just briefly, Domain Therapeutics is a French and Canadian company based in Strasbourg in France, in Montréal in Canada. And we have a focus on GPCRs in immuno-oncology, and we develop small molecule entities and new biological entities, antibodies, to address those unmet medical needs. So for GPCRs, as Michel mentioned in his introduction, they are quite an attractive drug target family. Almost 30%, 1/3 of current approved drugs, in fact, address GPCRs in a broad number of applications. So here it's a phylogenetic tree of GPCRs, some highlights of a few years ago of GPCRs that have been crystallized, but just the focus is on the different indications for which GPCRs modulators have been approved. So you see inflammation, metabolism, and others. As also what Michel mentioned in his introduction is quite unique that despite this huge implication in terms of providing drugs for medical needs, the application of GPCRs in immuno-oncology is quite weak still so far. So there are key targets in inflammation, autoimmune disease, and immunology. And for immuno-oncology, one of the key approach is to address GPCRs with antibodies. And one of the limitations was the ability to discover and move to clinic antibodies that are targeting GPCRs. I would say that the old approaches or still current approaches for the discovery of antibodies are still not working very well. And the classical approaches that have been used to generate antibodies against antigens usually direct binders that in fact recognize epitopes that are not really available in terms of biological applications. And so the different strategies currently applied and developed are based on the use of different types of antigens. Antigens can be peptides coming from the N-terminal part of or extracellular loops of GPCRs. They can be from cell lines that do overexpress the GPCRs of interest. They can be based on the receptors that are expressed and format in nanodisk or liposomes. Some companies now do work on the virus-like particle or exosomes. You have heard a nice presentation of Martine regarding exosomes. We can also use exosomes that are enriched particles for GPCRs to use them to discover antibodies. And of course, now some companies start to work on the RNA immunization. We all know the huge impact of RNA for COVID-19, and some CROs and biotech start to explore such a field to generate antibodies for GPCRs. From antigens regarding the strategy, so we can work with phage display, phage that will display antibodies. We can work with immunization of different species. We can work with rodent or llamas for the nanobodies, or even chicken. And we can also work with animals that have been humanized, especially the mice or sometimes the chicken, to immediately generate antibodies that are human from those species. In terms of analysis of the outcome, usually we work with classical methods, but now also one of the limitations was the number of hybridoma, for example, that can be screened. And some companies develop microfluidics apparatus to be able to screen hundreds of thousands of B cells to not omit quite precious producer of antibodies. And of course, binders are good, but functional binders are even better. So for most of those GPCRs, we are interested to have antibodies that will be antagonist, inverse agonist, or agonist, and binders will not be sufficient. It is important to be able to introduce functional readouts as soon as possible in the discovery and selection process of the antibody. And I will cover today some examples of different approaches followed for the discovery of antibodies for GPCRs. So one of the first approach that I'm going to present is based on the phage display of antibodies. Those technologies are followed by different companies like Distribute Bio, MapQ, or Proteogenics. What they do is they do express in recombinant manner GPCRs in fusion with green fluorescence protein. This will allow the people to follow the translated cells later on. They pre-incubate the phage display library with naive cells that do not overexpress the GPCRs to deplete non-specific binders at the surface of those cells. For example, if you do a recombinant cell lines with CHO or HEC293 cells, you are going to do a depletion of the library first on naive HEC293 or CHO cells to remove non-specific binders. And later on, you can incubate only with the recombinant cell lines, or you can continue to keep an equilibrium between the naive cell lines and the recombinant cell lines, the recombinant cell lines being detected thanks to the GFP. And you can do some wash and specifically sort by FACS the GFP positive cells, so the cells that do express the GPCRs, that will be eluted or co-eluted with the phage binding at their surface. And so after you can recover the phage through the lysis of the cells. The phage are resistant to treated lysis, whereas the cells are, of course, totally destroyed. And you can recover the phage, amplify the phage with E. coli, and perform this selection one more. And such a process is called panning, and you can do 3 to 4 panning. As soon as you have selected your phage of interest, of course, you can sequence the phage and recover the sequence and convert the sequence to IgG antibodies. And for example, here is an examples of data generated for a 6R5 receptor. So you see CHO transfected cell line. So you have the parental cell line, the non-expressing cell line, and you have also the CRTH2, which is a DP2 prostaglandin receptor, and the 6R5 cell line. And you can see that we can nicely isolate positive clones for the 6R5 receptor. The next step, of course, is to convert and confirm those data on cell lines that are more relevant and close to more T cells. So for that, we can work with UCAT transfected T cells, and here also to confirm that the binders previously identified on the cell line used for the panning were confirmed on those new recombinant targeted cells. Another approach is to have to work on more focused libraries, especially for peptidic GPCRs like chemokine receptors, as presented before, but also for receptors like from the complement and others. Such a strategy has been developed by Twist Biopharma. So they have created different types of libraries inspired by the biology of the receptors. So they have created library inspired from the sequence of the natural ligand of the GPCR, like from the chemokine. So they have worked on the structure of the chemokine and the binding site of the chemokine with the GPCR. Several X-ray structure or NMR structure have been solved. So they use those peptide and interaction sites to design specific peptides and create a diversity of peptides at such a place. And those peptides will be displayed at the surface of phage. And later on, we can use phage with this library, diversity library, focus library, for example, for chemokine, to screen for new binders, especially if we're interested to find agonist or antagonist of chemokine receptors. We can also work with other part of the receptor to guide the design of those focus libraries. We can work on the specific peptides from the extracellular loop or extracellular domain to create new binders that will favor or disrupt such type of interactions. And of course, when it is available, we can also work with the structure, the sequence and the structure of antibodies that have been previously developed for such type of GPCRs and create a diversity around the peptides that are involved in the binding site, for example, on the CDR, but potentially depending on how the discovery was previously made to create a diversity there. And a nice example from such diversity approach and phage display is for the GLP-1 receptor. Here you have different binders discovered through phage display, and you can see that those binders display quite nice activity in the nanomolar range, and all those antibodies, in fact, are agonists of the receptor. So it's not only they are not only binders, but also display pharmacological relevant functions. More classical approach are, of course, the hybridomas technology followed for GPCRs, for example, by companies like Mimabs or Synapse. In this case, mice will be immunized with peptides, with DNA, with RNA, with cell lines, and we are going to recover the B cells, and we are going to create a fusion of the B cells with myeloma cells through electrofusions to create hybridomas. and we'll put those hybridomas in a 384 well plate format, and we'll screen the supernatant of those hybridomas. So the hybridomas are going to secrete antibodies, and we are going to screen all those antibodies to see if they do recognize, usually by flow cytometry, the GPCR of interest that was used for the immunization. So as soon as you have your binders, so you do express your binders, you do several steps regarding the expression, the quantification, and the correct format through analysis of SDS page. You can later on see if those binders will bind to the human GPCR of interest. There are classical dose response that you can perform on the dedicated GPCR regarding the ligand. Of course, as we want to go to clinical trials, the tox studies are performed on cynomolgus monkeys. So it's important when we discover and develop antibodies against human target that they do cross cyno GPCRs. Is almost a request. Mouse is not requested, but cyno is. And from the binding, of course, the next step is really to confirm or to keep the functional activities. So here, example of functional activities obtained for one receptor for several of those antibodies. You can see that we can have antibodies that are quite potent in terms of inhibition in the nanomolar range or even in the subnanomolar range, picomolar range. This is what can be obtained for one receptor, but can be also obtained for second receptors. What I found quite elegant and back also to the original discoveries and the pharmacology of GPCRs is what has been set up by the company Abalone. They do work on with yeast. Yeast was for more younger researchers. It was used to characterize, discover the function, the pharmacology of GPCRs, the characterization of the first G proteins in terms of functions and so on. And in fact, they came back to this to those original work to discover antibodies. And what they do is that they do express on yeast some GPCRs that when the GPCR is expressed will induce, will keep the yeast to continue to grow. They have generated antibodies libraries, VHH, single chain heavy, Fab, or from naive or quite enriched. And what they can do is that they can screen and through co-expression billions of cells, and there will be one antibody per cell. So they can really assess one antibody per one antibody. What is interesting, for example, is when the GPCRs is only expressed without any activation, for example, we will have a death of the cells, so you will have no clones growing. If the yeast will, for example, produce an antibody that will be an agonist of the GPCRs, then the yeast will survive and will grow, and we are going to enrich only for yeast that do express agonists for these GPCRs. Of course, you can engineer in other manner just to select for antagonists with a revert system. I think it's quite interesting. It's quite straightforward, and you have a direct selection of functional antibodies. And it's also back to original work on GPCRs 20 years ago. And I think that using again such a strategy, but for antibodies, is quite promising. And of course, when you have all those clones selected, you can perform single cell sequencing, but you can also perform next generation sequencing and see the enrichment of the sequence and to select the most promising binders from the whole library. In the introduction, I mentioned also the microfluidic. What is quite painful when you do work on the discovery of GPCRs antibodies is that you can immunize animals, you can do titration on the serum, you can detect very nice and high titers in the serum of the mice. But unfortunately, when you start to screen the hybridoma, you cannot sometimes find good binders. We know that the mice or the chicken or the rodent do express the good antibody, but you cannot come back to the B cell or B cells that have produced such an antibody, and it's quite frustrating. So based on these observations, some companies do work on the options to be able to screen hundreds of thousands of B cells and try to not omit and the producing ones. So we can work with mice that have been immunized. Some companies also do work with serum from patients that, for example, will produce autoantibody against GPCRs that will lead to autoimmune disease. Work with isolation of those B cells, work with microfluidic chips, do encapsulation of single B cells in droplet, and can add in the droplets some reporter system to see if the antibody that are going to be produced by the B cells will activate or inhibit the GPCR of interest. And when an antibody that will be agonist is detected, there will be we can detect a fluorescent signal. And when we analyze those droplets in an individual manner, like by flow, and similar to the flow cytometry, we can select and enrich only for droplets that are positive and discard the non-positive ones. And after focus on the B cells here, do as before, deep sequencing and analyze the sequence of the antibodies. For example, when you do work with a company like VLabs, they can generate almost 1,000 samples per second in terms of analysis. So in more concrete manner, how it is performed, so you have a droplet. In this droplet, you have different entrants. So you have the B cells. You can have the GPCR that express the cell lines that do express the GPCRs. When the GPCR will be activated, you can have a reporter system like fluorescent, like calcium flux or things like that, that will induce fluorescent signal about binding of the antibody. And so you have all those components in the same droplet. And as I said before, you analyze through the microfluidic system and through a laser and detector to enrich only for the B cell that do produce agonistic antibody for the GPCR of interest. A company based in Canada called Abcelera has performed a lot of work before in academic world, before to start to be a biotech, and now quite a successful biotech with a recent IPO. What they do is they are working also with microfluidics. They are working with chambers, small chambers. In fact, in each of those chambers, you have only 1 nanoliter of volume, and the chambers are from 100 to 150,000. So here it's an example of previous generation. And in this chamber, you can put B cells that are going to produce antibodies, but through the microfluidic system, you can add, for example, reporter cell lines, target of interest, counter cell line, other cell lines that do express the cyno or the mouse receptors, to see if the antibodies that are going to be produced are selective or not from a receptor family or will cross-react among species. So the small volume enabled the secretion of B cells, because in 1 nanoliter you will have 1 nanomolar of antibodies quite rapidly. The support in terms of interrogation of the microfluidic chambers are based on the microscopy studies. The apparatus is designed in a sense that you can really deliver the different ingredients step by step. As said, the device is more than 150 chambers, and you can screen for more than 1 million B cells per day. So it's different from hybridoma that need several days, and usually you screen from 1,000 to 3,000 from 1,000 to 3,000 B cells. Here you can really screen more than 1 million B cells. So the starting material are based on the usually B cells. You can have them after immunization with peptide, with DNA, RNA, or even recombinant cell lines. You recover the B cells that are going to produce the antibody. You will have, for example, the parental cell line as a negative control. You will have your target of interest in this case. And for example, another cell line that do express a GPCR that is homolog, but you don't want to address. For example, another receptor from the same family. You can pre-label those different cell population with different dyes that will allow you later on to address and to find which cells do express which type of receptors. And in the medium, you are going to add also a secondary antibody that is going to be labeled with a fluorescent dye. And after you can address and interrogate if an antibody will bind to a receptor thanks to the secondary antibody and thanks to the dye within the cells, if the antibody will be specific for one cell, meaning for this GPCR of interest versus the others. And if it's the case, you do recover the B cells and you do the sequencing. So in concrete manner, how it looks like. Here it's an example of microscopy images that you can obtain with a bright field, with a passive dyes. You have the mask, and you can see if you have an immunoglobulin that will bind to a specific target of interest, in this case, the target A only versus the others. Of course, you can also detect antibodies that will not be selective and that are going to cross-react against 3 antigens. So you can do really multiplex screening for specificity and potentially cross-reactivity. So I wanted to try to provide some examples. At domain, we do follow some of them. Why several in parallel is because they are quite challenging, quite complex, and sometimes some strategies are working for one GPCR of interest, but not for the others. So we have to maximize the chance of success working with one or several of those technologies. Regarding the GPCRs, of course, the interest is quite huge for autoimmune disease and in immuno-oncology. I think till today they are quite still challenging in terms of discovery of for the discovery of antibodies, especially as we are looking in most of the case for functional antibodies. So GPCRs are being addressed in the past for small molecule entities with inverse agonist, antagonist, or agonist. We would like, for most of the cases, the same type of pharmacology. So binders might not be sufficient, and we should really address functionality. You see that some of the technologies I presented to you before, people working on such technology try to address the pharmacology as soon as possible from the discovery or during the discovery process. From historical established approaches, you see that new approaches are really developed and with specific application for membrane proteins and especially for GPCRs. Based on those new approaches, there's a dramatic increase of options and chance of successes, of course. When we see that, for example, at AbCellera, they can screen more than 1 million B cells and on chips that are with more than 150,000 wells, of course, we can say that now the screening of B cells can be almost equivalent to HTS for small molecule entities, which was not the case until really recently. It was not the case before the last 2 years. the future is quite bright. From our discoveries, from our competitors also, there are several breakthrough data that are at the corner. Not going in details, we do work on programs for which we can, in monotherapy with antibodies against GPCRs, we can cure in monotherapy 100% of the mice in models that are not working for immune checkpoint treatment. So it's very amazing, and thanks to the GPCRs, and I think a lot of companies now are moving in the field and will continue to enrich the field. And the immuno-oncology, of course, soon and already now will profit from all those new devices and tools that are developed. And based on that, I will conclude, and I would like to thank the organizers for the nice symposium, and I will be happy to pick up any questions.
Thank you very much, Xavier, for this nice review of all of what's happening in the field of generating antibodies against GPCRs, and in particular functional antibodies against GPCRs. Maybe I will start. I see that questions are coming up on the chat, but I will start by a question. So you're right in saying that traditionally GPCRs have been difficult to work with in terms of generating specific antibodies, and the field has been plagued with this for a couple of decades, right? It's been very difficult to get good antibodies, and it's sort of delayed the field in some aspects. Now with all of this activities happening, I mean, there's all of these technologies, all of these approaches, I stopped counting the number of companies generating antibodies against GPCRs. It's a very large number. So do you have a sense of what is the coverage right now for functional antibodies? And when I'm saying antibodies here, I mean intrabodies, nanobodies, all of these. You have an idea of what is the coverage on the 400 non-olfactory receptor? Do you think that we have functional antibodies for, a handful, for, a sizable proportion? What is your view on this? Because you follow this literature quite closely.
Yeah, functional antibodies it's really a challenge. Usually people are still working with to discover binders, classical approaches, binders. And after from the binders, they try to narrow on the functionality and the pharmacology. You know that thanks to your work, we have a platform in Montreal. Several biotechs do address to us their antibodies for functional characterization because they have not been discovered through a process where the function was part of the process, and they have to screen after for functions, and only few of them or none of them, in fact, are functional antibodies. So it's really a big challenge, and I try to focus on few of them where you see that potentially the functionality can be addressed earlier. After, if we go in more techniques, what Martine has presented is quite interesting. You see that she discovered, and others have discovered, and she had such nanobodies that then can lock GPCRs in a defined conformation. And then, of course, you increase the chance to discover antibodies that will recognize a lock conformation, lock meaning agonist or antagonist conformation. And this is a way also to potentially have antibodies that will be relevant in terms of pharmacology. But it's always needed, so far, it always needs a dedicated technology pre-existing with you have first to discover a first antibody or nanobody to lock before to discover the second one, or you have to engineer a microfluidic system to address that in real time, and it's quite challenging still.
Okay.
There are a lot of companies, but the success
still to come, right? So yeah. So Alex is again commenting on the very nice overview that you gave, and he was wondering, he was wondering, which GPCR target space you think is particularly relevant for functional monoclonal antibodies over other compound types. Do you think there's special cases of GPCRs that would be more amenable or be preferred to go with monoclonal antibodies? And he's talking in particular to agonistic monoclonal antibodies. But you can answer it in a general sense, and then maybe a few words on the agonistic. But
yeah, the question from Alex was more on which type of GPCRs are more favorable.
Yeah.
for agonistic and So
well, for compared to other type of compounds, you know, which GPCR should we focus our attention for going for antibodies rather than small molecules? That's the way I understand it.
Two aspects. The first one, where antibodies will be unique and where small molecules are irrelevant, are antibodies for depletion. So GPCRs are quite of interest for when they are specifically expressed on tumors and not in or overexpressed in tumors and not in normal populations. Then to have antibodies, binders that will only will bind to these GPCRs with a depletion function, thanks to the FC with ADCC, CDC, ADCP will be good, and small molecules will not do the job. For the antibodies, antagonist or agonist, it's where people, for example, for the antagonist, it's where people have developed competitive orthosteric small molecules that are not good enough. Of course, we do screen also to find allosteric to be, in a sense, resistance to the ligand and the concentration of the ligand. It's not always easy. And there, potentially, antibodies, antagonistic antibodies can do the job. In the same manner, Martine presented for chemokine receptors. Several one GPCRs can recognize several chemokine receptors, meaning different binding sites and so on. You can potentially have nice small molecule entities if they are allosteric to block several or most of those peptides, chemokines. But we do consider that potentially for such type of GPCRs, antibodies that will lock the GPCRs in an antagonistic manner will prevent most or all the chemokine to bind. And the third answer is more for the agonist. Clearly, for peptidic receptors, it's why I mentioned GLP-1. I will say that for glucagon, GLP-1, and others, where you do consider that having an agonist is good, small molecules sometimes do not do the job because the peptides are quite big, and to have a small molecule going in the orthosteric site and be able to activate the receptor is not sufficient. Peptide stability and so on is quite challenging. Antibodies can be a nice alternative, and I think it's where they should be for the agonistic functions.
I fully agree with you, and I would even say that for the agonist, the peptide receptors, I would even say, especially class B receptor, which have been traditionally very difficult to drug with small molecules.
Yeah.
Joe Mancini is asking if you could tell us a bit, you know, which receptors are well advanced into the clinic with therapeutic antibodies, and you know, in your mind, which ones are the most advanced in the clinic? If you have a perspective on this.
You have mentioned there's one antibody approved, which was on 6R4. There are others, but for migraines, which was quite challenging at the beginning to think that we are going away from onco and immuno-onco. I cannot really mention several targets, but we and the others, and you can go through our website, we do work on the GPCR of interest, and others are still at the research stage. For example, when people have developed small molecule entities for adenosine receptor, other companies now are developing antibodies in immuno-onco to address adenosine receptor. With antagonists, especially because the concentration in the tumor microenvironment of adenosine is quite high, and the small molecule sometimes cannot compete with very high micromolar concentration of agonist. Antibody can do the job. So such type of receptors are the front runner. The one that now are moving on, for example, are on the complement, C5a receptor. First for COVID-19 from Innate Pharma in Marseille. It was in onco and in COVID-19 due to the crisis. They focus on COVID-19, but MorphoSys and IMAB, and IMAB China now are in clinical trials in immuno-oncology with an antibody against C5a receptor, antagonist C5a receptor in the US in immuno-onco.
Thank
and
you.
The new ones will be mainly on chemokine and peptidic receptors. So some that Martine mentioned and others.
I was going to say maybe some of Martine antibody or antibodies will move forward in the clinic soon, hopefully.
Yeah, I wanted to say that. Yes.
So I think the one you're referring to, Xavier, in terms of migraine, I think is a calcitonin receptor.
Yeah, exactly.
Antibody that has moved into the clinic.
It's
approved. Yeah. William Wu is asking, if structural biology can be of any help in guiding the interactions between the antibodies and the receptors, or whether there's some headways into that direction.
Yeah, it is useful, and in fact it is needed. It is needed at the end because to patent antibodies in the speaking in terms of industrial and applications for patients, in the US you should provide the paratop and epitope antigens antibody and the binding site. And of course, to provide, because it's what you protect the sequence of the antibody, but you have to protect also which part of the GPCRs is recognized by your antibody, meaning that you have to understand that. It means that you have to explore and improve the binding site to have the best antibody for such a part. So it's very, very, very useful. There's a company like Calixar and others that produce for example, soluble form of A2A receptor that can be really used after with the structure to really interrogate the binding site of the antibody and with the receptor. So for me, it will be very important. Of course, you need previous structure to guide you, and you see that it's also what Twist is doing with their focus library. Or you have to start from scratch, but you need also other types of tools. So it's quite still easy. It's not like that. It's more challenging than from small molecules. Small molecule, we can do scaffold doping. We can improve molecules based on the previous information. But due to the limitation of the information on the epitope, paratop from US authorities, for the patent, if you follow such a process, you are going to fall down in the same pocket, and your patent might not be granted later on. So for us, we have to find other strategies.
Thank you very much. So I will acknowledge two other questions before we go to the break. So Yuri Saragovi is asking whether, based on what we know about RTKs, whether people have targeted the glycosylation of GPCRs to generate antibodies.
What I can say, I think people from my team will laugh about that. Yeah, we do work on the we can work with proteins that are synthetic or in recombinant manner, meaning that the translational modifications will be different. And we do know that those different sites of modification, glycosylation, sulfonation will be key factors, meaning that some strategies are not going to deliver because of those specific modifications that are real, but not addressed with the classical strategies. So we learn that in an indirect manner from lack of success to understand why, and after we start to understand why.
Okay,
so,
last question.
Yes.
Okay. Last question I will allow is that from Jean Labrec, who's asking, what is the contribution of affinity maturation? In other words, sort of lead optimization in terms of engineering the antibodies.
Sure. Usually from phage display, we have to perform affinity maturation. So as soon as we have the sequence of the different antibodies, we introduce site-directed mutagenesis or random mutagenesis, and we perform new phage display with the diversity that come from the first binders. From immunization, mice, for example, then it's more the humanization. It had occurred that we have even sometimes to perform a reverse engineering, meaning that we do not have to perform affinity maturation to increase the maturation, but sometimes we have to work on that to decrease the affinity. So I will say that it's mainly for the phage display, for the immunization also, but it's less stringent, and the impact is not so important.
Again, thank you very, very much, Xavier, for this very nice tour of where we stand with this, and also for a very active question period where a number of insights were discussed. So as I mentioned in my introduction, I think Silvio has been the first person really pushing the agenda of GPCR in oncology at a time where most people were interested in oncology and cancer were thinking in terms of oncogenes only and mutation. And this was before mutations were really found in GPCRs and well, G proteins were starting. But you know, it's really the one that pushed the envelope to make people aware that because GPCRs are regulators of so many processes, and that because they are good drug targets, they should be considered more into this field. And clearly, his voice has now been heard. And as I mentioned at the beginning of the talk, he's written a number of very influential reviews on the topic. And I'm very happy that we have him to sort of close the presentation part of this symposium today. So Silvio will present the talk entitled Mutational Landscape of G Proteins and G Protein Coupled Receptors in Cancer, New Multimodal Precision Cancer Therapies. Silvio, take it away.
So thank you, Michel, for the kind introduction. And of course, it will be much nicer to be all together in the same place, hopefully next time. So I will try to...
Yeah, I just need to stop sharing my screen. It will take a second.
Yeah.
Here we go.
Perfect. So thanks, everybody. Thanks, the audience. And certainly thanks, Michel, and all of the prior speakers. I will be able to follow your, your presentations in the recording afterwards. So let me share this, my screen number 1. Sure. 1 second. And this. I hope you can see me. Perfect.
Yeah, we see you. We see your screen. Fine, thank you.
Perfect. So what we'll try to cover today. So thanks, Michel. So this is precisely what I will try to cover today. So first, the G proteins and GPCRs as oncogenes that were, shall I say, neglected for some time. Oncogenic signaling networks, I will just briefly touch on that. You will see in very specific context. In the most recent use of computational biology approaches, computational data set analysis, etc., to reveal what we call the human onco-GPCRome, and then focusing specifically on G-alpha-Q, we call the GNAQ oncogene, and going from the bench to the clinic. And I will mention at the end, so the other speakers have presented this on what we call the oncocrine signaling by GPCRs, so normal GPCRs in the context of the tumor microenvironment. And finally, I will just touch with one slide what we think part of the future is, which is the focus on GPCRs in cancer immune therapies, an area that we are working a lot in some of your labs as well. So first, my disclosures. I don't think they influence any. In going to what Michel said, so the GPCR, the first study on GPCRs in cancer was really in the 80s. There was the time of the discovery of the oncogenes. So you take DNA from a tumor, you grind it, obtain DNA, transfect into cells, and identify using these focus formation assays, you can identify transforming DNAs or oncogenes, and you sequence and you can publish in Cell. Okay, that was the old good days. So it's certainly you need to do much more right now. This particular study, perhaps the first one describing one GPCR, the mouse oncogene, as transforming, was really not followed too much because precisely there were no mutations. So that was the key issue. There were no mutations with respect to the normal mass receptor, and that was viewed as, you can call it. You can call it a cloning artifact, or you can call it something that happens, but it was not really related to cancer. That's what we started working on this particular topic in the 90s, and it showed that GPCRs using masculine receptors can be ligand-dependent oncogenes. Activating mutations can make it active. Other labs as well. G proteins themselves can be oncogenic. We'll be describing some of these today, especially with GQ. And then late in the 90s, our lab and several, and Martine, of course, she has been leading this aspect. We focus on viral GPCR, so GPCRs that are expressing viruses, with emphasis in our case in the Kaposi sarcoma virus, which encodes one particular receptor within the open reading frame 74 that is, we believe, and which provided evidence that maybe the transforming oncogene within the Kaposi sarcoma virus. Although Martine was showing, so that it's possible to target them with nanobodies. They are not direct targets for this viral GPCR, but that was used in our case and then confirmed the clinic that you can target the downstream pathways from this GPCR. Specifically, mTOR is what we focus on, and rapamycin was used for treating, in our case, mice. But it was the first demonstration, really, rapamycin can be used to treat a human malignancy. So KS was the first one. Renal carcinoma is where it was approved, but the KS was the first disease. I'm using this as an example of you can target downstream pathways similar to what it was presented right now. Even if you cannot target all of the GPCR, they activate that pathway. That can be one mechanism to target the pathway. So the surprise really happens when working with our colleagues in Genentech and when I was still at NIH working with Tracy Handel here at UCSD, is that when sequencing the cancer genomes, so all of these efforts, TCGA and prior to that, sequencing of cancer genomes, and start looking into mutations in GPCR, what we found more than we expected. Roughly 20% of all cancers have mutations in GPCRs. That was much more, again, than we expected. And then the question is, are there drivers of their passenger mutations? They may have some importance, of course, but they're not the ones that drive the cancer. And still, it's an issue. I will address a little bit of that. But we found many hotspot mutations that make G protein alpha-7 is constitutively active. And right now, most cancer centers, more solid tumors, they will be sequenced. And there's usually 400 gene panel is being used for sequencing. 7 of those 400 genes are GPCRs or G proteins, which is really, really exciting. So you can say that G proteins and GPCRs are now really in the radar of all medical oncologists. The only key issue you need to remember, you need to call by the gene name. So if you call it G alpha Q, nobody will understand what you're talking about. We know what you are saying, of course, but it will, in the medical field, it's called GNAQ or GNAC, just to give you an idea. So call it by the name of the gene, and you're fine. And then you know what you're talking. So more recently, they this again. So I try to share. This is published, but sharing some elements that you may not be aware or may not see that in the paper. So when we did analysis, all of the GPCRs mutated cancer, so we did the reanalysis and then focused on all of the cancer types in which these GPCRs are mutated. There are some surprises. The vast majority of the GPCRs are mutated in the GI tract. So either they are mutated in colorectal cancer, stomach, or pancreas. And that's quite exciting, because other cancers like melanoma and many other cancers, they have much more higher mutational rate. And yet most of the GPCRs are all around the GI tract, which is very, very exciting. With a few exceptions, I will refer it in the case of uveal melanoma. So what are all of these mutations? Are they random? The answer is probably no. First, because in other cancers that are heavily mutated, you don't see them, or you see very few. This is using, of course, if you go deeper, there are many mutations. This is using bioinformatic tools or pipelines that reveal those genes that are mutated above what you consider the basal mutational rate of that particular tissue. It's tissue by tissue, patient by patient. So this was, so we can say all of the mutations are above that background or threshold, and yet the vast majority will be only in very few organs. And if you put all together this in collaboration with Aska and Francesco Raimondi, so we were able to map all of this to the canonical structure of the GPCR. And that's really exciting, because if it's random, it will be anywhere, but in reality, the vast majority are clustered about a very, very small subset of structural motif, so that you can identify NPXY motif, DRY motif, etc. So they're all clustered in different places. And using a paper we published recently with Aska and Francesco. So what we show, we can predict what will be the consequences. We have not proven. That's something that many of you may be doing or want to do. So what we did is to predict what will be the consequences. And the most important consequence, perhaps, is activation of the GI pathway, of the GS pathway or the PKA signaling pathway. So the vast majority would be loss of function in GI GPCRs or gain of function in GS GPCRs. There are also some GQ here and there, okay, so, and also some G13. But that would be the take-home message when you take globally. And if that makes sense, there are many mutations in G protein now, G-alpha-S, mutated in colorectal cancer, appendix, pancreatic cancer, and many adenomas. So we see the GS pathway becoming a little bit dominant in many of these tissues. In addressing something that was just mentioned before, long time ago, we provided evidence that may be part of the GS pathway, may be part of a you can call a paracrine or autocrine activation, mostly paracrine, through the COX-2 release of prostaglandins, particularly in the case of colon cancer. In a study that bioinformatics and structural biology approach is coming soon, what we show is that in particular for some cancers, colon cancer will be one of them, there are many non-overlapping mutations or genetic alterations in the GS pathway. So this was done by Dana Stephen, so she's getting her PhD in a couple of weeks. So that's excellent. So going to this, the one I will focus will be in G-alpha-Q, specifically G-alpha-Q and GNA11, or the receptor CYL-STR2, which is a GQ kappa receptors. They are mutated primarily in uveal melanoma. So going back to the future, if you will, that was our first paper from our lab in which I was the senior author. So for many of the junior members of the audience, when you transition from being a postdoc to have your own lab in your tenure track, that was precisely the first paper in that we show the G-alpha-Q is mutated. And certainly the work by Boris Bastian is what showed the G-alpha-Q is mutated primarily in uveal melanoma. That was really exciting. So uveal melanoma is different. So there are around 2,000 to 3,000 cases every year. They don't have BRAF mutations, typical from skin melanoma or NRAS. They usually fail to most immune therapies. Now, 92% have G-alpha-Q mutations. I would just also mention skin melanoma, 4% have G-alpha-Q mutations as well. And it's unfortunately 50% of the uveal melanoma patients will develop metastasis, and when they develop metastasis, usually it's lethal. So within 5-6 months, sometimes 1 year, so the patients will succumb to the disease. There are no current therapies for uveal melanoma. Most attempts have failed. So in going back to the oncogenes, you can clearly see it's a GQ-driven cancer. There's no question about it. So 50% will have G-alpha-Q mutations, G11 will be 45%, around 3-4% will have CYS-TLR2, which is GQ kappa receptors. There are also a few other mutations. So it's a cancer with very few mutations. The other mutations are in splicing factors, molecules involved in splicing, or BAP1, which is an epigenetic modifier, which are not really involved in the initiation of the cancer, but it's important. Mutations in BAP1 usually lead to metastasis. So that would be the metastatic gene, if you will. But the driver, again, is G-alpha-Q. Attempts to block the MEK pathway, canonical, classical GNAQ, PLC, ERK pathway did not work in the clinic. So although it activates ERK, if you use MEK inhibitors or any other inhibitors of the pathway different from melanoma, it will not be responding. So there is no increase in survival, not increase in of the patients. So I just using, so this was recently published, so we did a combination of the, again, now analyzing the driver oncogenes that was publishing as a review in FEBS a few weeks ago. So what we show is that just reflecting here, the vast majority will be one single residue, 209, very few cases 183, that will lead to constitutively activated G-alpha-Q. And so that's very clear. It's a single residue, similar to RAS. There are very few residues that are mutated. This would be like a canonical, classical oncogene. And so I will go very briefly this. These are some old studies to put some context to the most recent. So trying to understand the classical pathways that we are aware of, PLC, knowing that they may not be the only ones
that contribute to this disease. So we were back to, so we did very, shall I say, simple analysis using, as a reporter, AP1. We know the AP1 was activated by G-alpha-Q. That was studied many, many years ago, and some of you as well in the audience. And what we did is screen in Drosophila cells in Harvard using dsRNAs against the entire genome, Drosophila genome, to identify what mediates the activation of AP1 from GQ-kappa receptors. And we identified many members of the Jun kinase pathway as we expected, Jun, FOS, main transcription factors. We did not identify PLC or RAS or any of the ERK pathway, but very, very exciting, we identified components of the trio and also of the Hippo pathway. That was very, very unexpected. So what is trio? Trio is a guanine nucleotide exchange factor that activates RAW, and then working partially in collaboration with John Tesmer. So we have a crystal structure now of GQ bound to trio. It's this very, very high affinity interaction. So it's when GQ is activated, we'll in a two-step process, we'll recruit trio to the membrane, will activate trio. At the same time, this interaction is relatively stable. You can even co-IP. And we see very, very nice that through trio, it induces activation of RAW. The interesting, if you knock down trio, so trio first is overexpressed in melanoma and many other cancers, you decrease tumor volume, you don't affect the PLC activation or the ERK activation, but you diminish tumor growth. And so going to the Hippo pathway, which has been much less studied or recently, shall I say, recently identified a few years ago. We are lucky here at UCSD, we have Kuliang Wan, who is one of the leaders in the field. It's a pathway, it's very different from many other cancer pathways. It's a tumor suppressor pathway. So basically the activation of components of the Hippo kinase cascade in mammals with Drosophila will lead to a phosphorylation of co-activator called YAP or TAS. When phosphorylated, this pathway will induce phosphorylation, will exclude from the nucleus, and cannot bind transcription factors of the TIT family that are important for cell proliferation. So you can see, so these studies from DJ Pan and Kuliang, so they induce, for example, if you inactivate these kinases, liver will grow much more and it can lead to cancer. So making long story short, what we found using animal models, that G-alpha-Q can induce actually the Hippo pathway. You can detect by nuclear accumulation of YAP. You can see YAP in the nucleus using a transgenic combination of transgenic mice. You can see in human melanomas, if you knock down a YAP, reduces tumor growth very nicely. And we use, you can say, use a drug, repurpose a drug that is used for ophthalmology called verteporfin, which DJ Pan in a high throughput screen, they found they can block this interaction between TIT and YAP. And so you cannot use in the clinic, has many toxicities, can be used for only shortly, but it's very nice how it can be used for blocking this growth. So that's excellent. They gave us the cover of cancer cells, which is always cool. But the question is, can you target that? Again, so some of these, there are now new TIT, YAP inhibitors emerging in the some of which are being tested in the clinic, but we need to fix this quickly. So to approach this, we went back into a computational or you can call it systems biology approach. Nadia, so she's a brilliant graduate student, finishing probably next year. She will be looking for postdocs. So what we did in collaboration with Ethan Rupin at NIH and Pablo Tamayo here, we use something called synthetic lethal screens. So the synthetic lethal is something simple. So if you target the gene, in a normal cell, for example, gene A, the cells are viable. Target gene B, the cells are viable. If you target A, when B is altered in high expression, low expression, mutated, whatever, then it will be lethal. So that is a very important concept of synthetic lethality, because you expect that you may be able to target the cancer without affecting the normal tissues. That's really, really quite important. In this case, instead of using, like you often has been done using CRISPR screens and other, what we did is a computational, a new computational pipeline. Again, all of this is in silico, identifying genes that are expressed in the melanoma when they have a GQ, in cutaneous melanoma when they have GQ mutations, and that if they are ever reduced expression, then that lead to a better prognosis, for example, or when it's more expressed, becomes
a poor prognosis. So, and then using a combination of data sets, in vitro screens, large drug response screens. So there are many data sets that are readily available with the right computational tool. You can screen them. It's much faster than doing all of the analysis in your labs. And then finally, from this, selecting only those for which there are advanced drugs or already available in the clinic. When we did all of this, from all of the genes that you can imagine, we identified only 5 genes that qualify from all of this. The top one was a protein called FAK. The gene is called PTK2, for adhesion kinase, which is amplified in uveal melanoma. That was perhaps one of the reasons it showed up. And correlates very nicely with poor prognosis. So you can find this in the paper. It's very nice how it correlates. The beauty, there are many FAK inhibitors that have been advanced in the clinic. We are pharmacologists, so we run those responses curve. And if you take a number of uveal melanoma cells, they're very sensitive at doses that you can achieve in vivo in patients. And cutaneous melanoma are very insensitive. So if they have BRAF mutations, they're very insensitive. And so if you CRISPR FAK, the tumors don't grow. If you use an FAK inhibitor, very nice, the tumors also don't grow, or it has a dramatic effect, for example, proliferation. But the surprise, we didn't know how it works. So it's that when we did RNA sequencing, so I'm missing one panel here. When you do RNA sequencing, what we found that one of the targets, so we have very nice YAP conserved signature was missing. So what was affected? And indeed, if you use a FAK inhibitor, you see that YAP goes from the nucleus where it's active to outside the nucleus. So it's excluded from the nucleus using FAK inhibitors. Very nice. The important issue from this is that this information we also show that YAP was excluded also from the tissues in uveal melanoma in tumors, and based on this, a clinical trial was open specifically in MD Anderson, Miami, using FAK inhibitors. I'm missing one figure. I don't know how this appeared with the entire pathway. So the entire pathway is a little bit more complicated. The key issue is that what we see is that FAK specifically will phosphorylate one component of the canonical HIPPO pathway, leading to the inactivation of the HIPPO pathway, specifically the molecule called LATS, and the exclusion of YAP. So that would be the mechanistic aspect. So putting all this together, excellent clinical trial has been initiated using FAK inhibitors. We don't know the answer yet. But it very frequently, when you use a drug, a single agent, for example, BRAF inhibitors in cutaneous melanoma, so there is very rapid activation of what we usually refer to as a feedback loops. You can also call it compensatory mechanisms. For the practical purposes, these feedback loops reactivation or the activation of compensatory mechanisms can lead to drug resistance and at the end of the day, cancer relapse. So with that in mind, rather than embarking ourselves in a clinical trial, we decided to go one step further and now identify molecules that when blocked or deleted can enhance the response of FAK inhibitors in the context of G-alpha-Q. So that was basically what we were doing. This was done primarily by Justine Paradis. So she went back to France a few months ago. In Monica Acosta, so it's one of our students. She's undergrad, now she's enrolled in an MD PhD program in Cornell, so in New York. We're very proud of her. So what we did in this case, we did a screen, a CRISPR-Cas9 screen. We focus on kinases because we know kinases, there are many drugs that can be targeted, and because that makes the study much easier if you reduce the complexity of the study. We are doing right now, finishing the genome-wide screens. We're identifying very exciting targets, but this is really the goal was to identify something that we can bring to the clinic again. So we did a CRISPR screen. You use in this case, a kinome. You treat with vehicle or FAK inhibitors, and then you collect the DNAs. These are barcoded Cas9 expressing libraries, and then based on this, you can decode which are the genes we call dropouts, so that when you in the control cells will be the SG will have limited effect, but when you expose to FAK inhibitors, those genes will be removed. Those sgRNAs, suggesting that the cells cannot tolerate removing these DNAs. And so basically using these screens, what we found, perhaps not surprising, shall I say, the most important synthetic lethal interaction partners were all in the canonical or classical GQ pathway. So from PKC, components of the ERK pathway, so many of them were all in this pathway. And indeed, if you take FAK inhibitors themselves, and MEK inhibitors alone, they have some activity, of course, but when you combine them, they're really what's called synergistic. So the famous 1 + 1 = 11, they're really synergistic. And even better, alone, they will be cytostatic to some extent. If you combine, they induce very quickly, they induce apoptosis of the cancer cells. That's really cool. So then, bringing this to the tumors, so I'm showing just some of them. So in the tumors, what we see is that when you block FAK or MEK, in this case with trametinib, there is cytostatic, so the tumors, they don't grow. But when you use both combined, you see a decrease in the tumor volume very nicely. And the most important, you not only reduce proliferation, but you use the cell death as judged by the caspase activation of the cancer cells. Because the primary tumor is not the one that leads to a patient, affects the patient survival. So we developed some animal models for liver metastasis. So injecting cells with the GFP and luciferase in the spleen, you remove it, the spleen, so splenectomy, and then the cancer cells will colonize only the liver. So even if it goes everywhere, including the lungs, only the ones in the liver will survive. It's an issue that we are trying to investigate why is the case. You can see these liver mets, and we use the combination, so it's very effective in decreasing the tumor burden of these mice and enhance the survival. Based on this, we put together a clinical trial now. We call a signal transduction-based multimodal precision therapy. It's done in Jefferson University together with us. And the idea is that in this particular case, I did not show a lot of Western blots. So when you block with FAK, we see very quickly activation of ERK. And vice versa is also true. So when you block ERK, there's very quick activation, compensatory activation of FAK. So the bottom line is that you can affect you prevent this adaptive resistance, if you will, to one and the other. We call in our jargon, we call horizontal inhibition of compensatory pathways. So this is the idea, and using this approach, so we are already enrolling patients. I cannot disclose the outcome, but we're hoping this is going to work. So going one step backwards, if you will. So I mentioned to you about oncogenes and transforming pathways induced by G proteins and also by GPCRs. But I would want to remind everybody, especially for this audience, that it's not the only way in which GPCRs work in cancer. Very important, what it was mentioned before, we will call this oncogran, in general, we call oncogran activation of GPCRs. So basically GPCRs, if they're expressed in the cancer cells or other cells, so if they are expressed in the wrong place, at the wrong time, and they are exposed to excess production of the ligand, as was referring to in the transforming potential of perhaps the mass oncogene, or in our case, using GPCRs and carbachol with masculine receptor. So these receptors can play an important role in cancer. Keep in mind, so it's not the limitations. So we think very important angiogenesis is very well described. What we call these oncogran signalings, in which even the nerves, and there are several wonderful examples of publications on that, they release neurotransmitters, acetylcholine, noradrenaline, can influence the tumor microenvironment and induce activation of GPCRs that are expressed into the cancer cells. Very important for immune evasion. I will mention that specifically. And that's really very exciting opportunities to target GPCRs in cancer. And as was referred to, I have prostaglandin receptors, I need to include the thromboxane receptor, of course. This is a moving target. But in metastasis, they are very well known. Many GPCRs play very, very important role for metastasis. And finally, the area that we are moving aggressively into, we call the tumor immune GPCR repertoire. This is Vicky again. So together with Tracy Handel and our collaborators doing bioinformatics, we think there are many, many GPCRs that are expressed in many cancer cells, and some of which by bioinformatic analysis correlate with the ability to respond or not to immune therapies. And the idea is that the goals, you can target GPCRs or the regulatory networks to increase NK recruitment, increase dendritic cell recruitment. This is one area that we are very actively involved. Increase neoantigen presentation in the lymph nodes, and increase at the end increase the CD8 cytotoxic T cells effect into the cancer. So this is where we are going. Many of you probably as well. I think this is the final frontier. So summarizing this, GPCRs and GP proteins can act as oncogenes and are involved in human and viral situations. So human and viral oncogenes can be transforming. Deep sequencing of cancer genome, high frequency of mutations in G proteins and GPCRs in most tumor types. And now we see the emergence of G protein-driven cancers. In the idea that G protein, GPCRs, and then their downstream signaling circuitry, other classical, canonical second messengers, or non-canonical protein-protein interaction-driven can be exploited for development of new signaling selective precision cancer therapies. For uveal melanoma, elucidation of oncogenic signal circuitry with the trio raw AP1 YAP axis. Synthetic lethal interaction of GQ revealed the FAK can be a druggable target in modulating a YAP. And then synthetic lethal interactions now in uveal melanoma, they have GNQ with FAK, such as the FAK plus MEK can be in promising multimodal precision targeted therapy. And again, this is not only mutations on congruent GPCR signaling. Many wild-type GPCRs are druggable targets for prevention and treatment. And finally, the GPCRs may represent very exciting novel therapies for immune therapies. With that, I will just finish acknowledging everybody. In our lab, this was before COVID. We are beginning to take some pictures again with a mask in a couple of weeks without masks. And just thanking everybody and many of our collaborators around the world, some of which I have been able to mention. So thank you.
Thank you very much, Silvio, for this very nice overview of some fundamental concepts that you were involved in putting forward, but more recent data, very exciting data, and even data that led to some ongoing clinical trial. This is very exciting. We're eager to hear about the result of this trial in the near future. So I will open for question. I see a first one, I think, from Alexander Hauser. We also thank you for your talk. So he was asking about what is the main type of GPCR mutation, non-synonymous, introns, among the 20% that we find into cancer. And how does that compare with the mutation rate in other family of proteins?
So it's similar to kinases. So in principle, I would say similar to kinases. Most of the mutations are point mutations in affecting, so there are deletions, there are copy number loss. You can find, but the copy number losses, they don't correlate very well with the this disease, and they don't correlate very well and for example, mutations that will change the splicing areas. So they are all of the above, as you can imagine, but it's very enriched in point mutations, very rich in point mutations. So will be non-synonymous mutations will be enriched. And how do you compare? You can compare more or less with the kinases. So seem to be the preferred mutations in general.
So the question of Martine Smit, she's asking, why is it that most of the oncogenic GPCR mutations seems to be found into the GI tract disease?
Yeah. So to be practical, so the probably this is this is our working hypothesis, yet to be proven. So we think that the most why they are so rich in GI can be primarily because precisely the GS-PKA pathway. Seems like the GI tract is extremely sensitive to the GS-PKA pathway we call PKA-opathies. So if you, which some of the reviewers of our review didn't like it, they want to call it something else. So we call it G-alpha-S PKA signalopathies, just to make everybody happy. But the reality is, so if you looked into GI colon, using colon because it's one of the easiest to follow, there are 4% mutations in G-alpha-S. These are activated mutations, 201 C mutations, the vast majority. And then you see activation of PKA pathway, PKA itself, 1%, the C subunit A, C subunit beta, there will be also 1-2%, like 3-4% mutations in regulatory subunits. There are those that make it ineffective. Then many mutations in adenine cyclases. So those we cannot trace precisely. And there's around 7% of copy number loss, absolutely knockout of a phosphodiesterase 4D. So basically 7% of the colon cancers have no phosphodiesterase 4D specifically. So it's both alleles are lost. So bottom line, if you go through the entire pathway, so this will be part of this review that we put together with a lot of informatics, and structural predictions, etc. It's really remarkable. So, and that is also true, for example, pancreatic cancer. Everybody knows RAS is the most mutated. Around 12% of pancreatic cancer have G-alpha-S mutation. These are, again, the canonical 201C mutation, nothing special. And so, to some extent, we feel the pancreatic cancer as well. Stomach is a little more complicated because there are many etiologies, but that's the take-home message. So all the GI tract are usually use GS-coupled receptors for enhanced secretion, for example. They have mechanisms by which they are protected from any other effects, in this case cancer, but for some reason this protection is disabled in this particular, and they are very sensitive to this particular pathway. So in our case, we have developed animal models expressing for example, RAS, single copy RAS mutations. And our RAS is a very potent oncogene. As a single mutation will do very little, so the cells will senesce and will be eliminated. If you add a GS, which itself will make secretory, so for example, become what we call hypersecreting lesions. Many of the polyps, for example, in the intestine that are benign in nature, that will be around 8%, 10% will contain G-alpha-S mutations, for example. So they are hypersecreting, they become mucinous in nature, so basically have mucin secretion. But if you combine RAS and GNAS, it's fulminant. It's very quickly they will induce cancer. So RAS probably is the oncogene, and G-alpha-S will be the companion or whatever, the co-driver. But that's probably the case, why these tissues are very sensitive.
So thank you very much, Silvio. So there's a question from Morley Ollenberg. He's asking, from previous work that you've done showing that there was anti-cancer actions of metformin, is there a link between the action of metformin and some of the pathways that you described today, or is there anything that you've looked into this? So
the answer, so we are working right now, it's unpublished, please keep it, so this is open, I wouldn't say keep it confidential, but don't use in the clinic, so that's the most important. So what we think, so metformin can be very effective for the prevention of the metastasis of the uveal melanoma. So specifically, the uveal melanoma, I mentioned to you, 50% of the patients will develop metastasis. How do you know, how long does it take? 4, 5, 6 years. So if primary lesion is removed, it doesn't matter, the tumor will still occur because the liver is already seeded with the cancer cells. So what do you do in between? Nothing. There's not much you can do other than monitoring once every 6 months for the presence of metastasis. So one of the approaches is to identify something that will prevent these dormant tumors to progress. And so in that case, something very important, I mentioned to you this gene BAP1, PAP1, when mutated or deleted, which is around 50%, most of these patients will develop cancer. a metastasis. So the bottom line, if you have a uveal melanoma and you have BAP mutation, you are in very high risk. And so that's the idea, to identify something. And it's a very, very easy to identify subgroup of patients. They may benefit from anything. And so we did a drug screen with many, many drugs on one end. On the other, I mentioned to you that the uveal melanoma is very driven by the YAP pathway, which we, Hippo-YAP, which we target initiated by GQ. So when combined the screens for YAP, things that interact with YAP and things that can be used, metformin seem to be working excellent. Not when the cancer is there. You have limited activity. What we think is these cells that are sitting in the liver can be kept dormant. So you will find, so we are receiving a grant from the DOD to focus specifically on that. So you will see online very soon. That's very exciting. And how it works, besides the mTOR pathway, in which everybody focuses, including our lab, seem to be also blocking YAP. Precisely how it blocks YAP, this is, I cannot give you the details, it's under investigation, but it's a really, really cool and exciting mechanism. So the metformin component for the GPCRs, we are bringing specifically for the YAP-driven cancers in the context of uveal melanoma, but not for the treatment, but for the prevention of the fulminant metastatic disease in high-risk patients that have BAP1 mutations.
Thank you, Silvio. A question from Richard Leduc. And this, I think, needs to be taken into consideration with the fact that we have shown that GS activation mutations are found in some cancer as well. But his point is about, the differential regulation of the YAP pathway by receptors that are coupled to different G proteins. So the GQ, G1213, GI promote nuclear translocation of YAP, and GS does the contrary. So intuitively, you think, well, a GS agonist may be a good thing, but then on the other hand, you show that constant activation of GS is bad. So can you comment on this?
That is so very, very interesting and, shall I say, exciting questions. We do not have an answer, but we have an answer for the consequences. I mentioned to you about the pancreatic cancer. So that you have RAS mutations. There is MP53, and that leads to adenocarcinoma of the pancreas, which is fulminant. Usually, also the survival is very limited. 1 year, usually is no more than 1 year. So, I mentioned that 12% of pancreatic cancer have GNAS mutations. So, working with Anirban Maitra in MD Anderson, we developed a model. What we did that, so we have RAS alone, GNAS alone, and both combined, and then what happens? So what happens is this follows a completely different pathway for cancer development. They form tumors, they're very huge, but it fits into a different, so they are very secretory. They call IPMNs, so it's a long name. Usually they form like cysts, to make it simple, some of which will progress into cancer. And so then the question, why they have RAS mutation in GNAS don't progress into cancer is because precisely YAP is being blocked. Now you can call low grade, low risk cancer. I mean, this still is a tumor. But when the time passes, the regulation of YAP disappears. We don't know precisely how is the case. This is what we are investigating right now. And then it's when it becomes a cancer. So you have two pathways, and if you go to the cartoon, we depict precisely that. So pathway number one, typical RAS P53 mutations leading to adenocarcinoma, and then you have the GS, which I would say is even more frequent. GS RAS mutations, they will lead to these IPMNs, and YAP will be suppressed until this suppression disappears, it will progress. So precisely going back to what I was referring to Martine, so we think that there is something there that's preventing the cancer, the GI to have even more cancers, and probably the YAP will be that. But somehow, whatever connects one into the other, there are some publications, the PKA phosphorylate-directed LATs, we cannot see that. I can tell you that it's a little bit more complicated than we expected, this particular component, but sooner or later, this you can call tumor suppressive mechanism can be deleted, or you can override that, leading to cancer. So the answer is complicated, but yes, that's extremely important. And if we identify what prevents the precise mechanism by which YAP is being blocked, we may be able to identify who is at higher risk of progressing.
It'd be interesting to see if there's a differentiation between the downstream, effectors being involved in this. In terms of, is it really cyclase, or is it something else that is involved in the other pathway? I mean, that's something that is, I think, would be worth looking into.
What is exciting is also tissue specific. So going back to the activation, then the same is true. G alpha Q is mutated. Probably some of you know, is mutated very heavily in. I mean, it's the driver of these lesions. Many children, many kids, they are born, they have these red spots. It's called port wine spots. 1 every 100, 1 every 200 kid will be born with some red spots, and then they disappear afterwards. So these are GQ mutations. The majority are GQ 183, so the one that blocks RGS interaction, just to give you an idea. And so that mutation is very specific, and usually the cells will senesce, will not progress. Some may proliferate and form something called Sturge-Weber syndrome that is driven by GQ as well, in which hopefully some of the work we are doing can help. But very few, these mutations in GQ can be found in many other places, and usually it's not oncogenic because the cells will undergo senescence. This was also the susceptibility of some tissues to certain pathways. How the cells, using our jargon, so that you can say tissue-specific bias for who will be responding one way or the other. And the GI is prone to respond to GS. Most other tissues are not.
Okay, so I will allow only 2 additional questions. There are others, but people will be free to go to the breakout room after. So Brian Ari is asking, did you pick up any mutations in the GRK and G protein receptor kinases in your analysis? So
we did not include. So we have done more recently. So of course, in Michel, so he did the analysis of the RGSs. The answer is yes. So what we do not find is hotspot mutations. So that's for cancer, people working in cancer, they're always call it an obsession to identify hotspot mutations, because that is an indication. It's like the smoking gun that something's really, really happening. That's why with G alpha Q or GNAQ 11, GNAS. So these are hotspot mutations. These are 209 or 183, mostly 209 for GQ, 201 for GNAS. There's no question about it, and you can predict what they do. If you don't have hotspot mutations, can become more complicated. We did not find hotspot mutations. This doesn't mean that in a complementary fashion they can be affected. So that I think is very important. There are RGS mutations. They are very likely GRKs. I cannot tell you. So we collected information, but if anyone has interest, happy to share all the information, certainly.
I'll try to do 2 if we can do it quick, because there's only 2 left, so I don't want to leave somebody out. So question from Yuri Saragovi, uveal melanomas are considered cold tumors. You mentioned that they don't respond to immunotherapy. So the short answer is, did you look at combination with inhibiting the pathways you've looked at and immunotherapy? So is the resistance to immune response come from these pathways?
So the answer to that is probably, the answer is probably will be tested, because FAK now is being used in combination with immune therapies to diminish the fibrosis of the tissues in other cancers. It's a very reasonable approach. You don't know until you try. We have developed in our recent paper, we use that. We have developed syngeneic models of uveal melanoma that we can use to test that. The answer is we have not tested. That said, the prediction is likely that it's not going to work, because this is one of the few cancers that have very, very low mutational burden. So there are very few new antigens available. And usually there's a very nice correlation between mutational burden and response to immune therapy due to the presence of new antigens. So this looks like a pediatric cancer. They have 5 or 6 mutations primarily. This one, so it's very likely it will not work, but that's why we develop these models. It needs to be tested. Thank you for that.
Last question from Shane. Whether or not, to confirm the role of the GQ pathway, if you've tried the known GQ inhibitors that are out there, and whether or not, and there's more recent ones with the improve pharmacokinetics. So is there anything that you've looked at?
Yeah, we were involved, so that we were not the primary drivers with the AV Costeni, so we involved one of the papers using this GQ. So I shall say, I think that we were the first ones to put into mouse. Okay, the drugs were there, but you needed to do the experiment, and that required AV's lab to get a lot of this drug, because it required a lot of. So the answer to that is yes, we try. Yes, we published in concomitant with a few other papers. It was published like 2 years ago. The answer is that this, as far as I can tell, the drugs are a little bit toxic. So the therapeutic, what we call therapeutic index. So you can cure the cancer, but you can kill the patient. So that said, in cancer, so the cancer will be gone, okay? But this was, so in this hypotension and many other things. So, but the reality is that it all comes down to now a pharmacokinetics and certainly improve delivery, but also the possibility to target only to the cancer cells. There are many ways to take a drug that is likely to be active but toxic, and make it less toxic, and make it more targeted to the uveal melanoma. So that's will be the way to go. Happy if anyone is in a position to do that, we can help you.
Or targeting the right pathway, right? Targeting maybe the trio P63 or the Rho pathway rather than targeting the PLC pathway, right? Maybe that's also something that can be looked at.
So we're doing this with John Tesmer. So we get into hopefully soon the structure using cryo-EM of the trio bound to GQ and the components. We think the trio, so we published this in the trio itself. So it's a little bit unusual. The ability to open and close in the DH domains is the one that affects the Rho. It's a little bit unusual because they are usually in an inactive form, but it's like a conformation in which two different domains that touch each other. So we think that a possibility is not preventing the GQ, which would be ideal, but the possibility is to identify a drug that will keep this close. So that is where we are targeting our efforts to identify something that may prevent the activation. The initial binding with GQ is like a 2 steps. So it seems like the initial binding is a low affinity interaction, but then once it binds, it will be like the Cas-Fly type of interaction in which it will be wrapping up GQ. So once it's formed, it's probably very high affinity interaction. So that may make it difficult or maybe more druggable. So this is someone else's expertise, but again, happy to help if anyone has some ideas.
Okay, great. We should talk about this because I think there's some interest into developing some of these more pathway selective inhibitors of G protein. So for those of you who were not, the questions were not translated to or transmitted to Silvio, you will be allowed to go to the breakout rooms in a few seconds. And I will keep my closing remarks very short so that not to take too much time from the people going into the breakout rooms, where we've tried to mimic more or less what happens in a real meeting, which is that after talks, people congregate at the front of the room, talking with the different speakers of the session, and this makes very active and interesting discussions. And to make them more free, this part will not be recorded. So I will encourage every of you that want to talk with one of the people to go there, and a bit later Louise will come in and explain to you how to get in the breaking rooms and move from one to the other. So first of all, I would like to thank the speakers, because I think you will agree with me that these were 4 fantastic talks that really drove the message across that I think GPCRs are important targets for drug discovery in the cancer field. I think the evidence for their role is now overwhelming. We have the tools, many sets of tools were described today, the antibodies, the nanobodies, the ability to screen for the activity of these compounds, and as Silvio mentioned, a number of clinical trials are already going. So I think this number of 8 drugs being used, 8 GPCR targeting drugs being used in cancer, I predict, and I know it's tough to predict the future, but I still predict that in a few years, this number will be multiplied by a big factor, and we'll have a number of drugs approved for therapy, and in particular in combination. I think combination therapy may be a very important aspect for the future of cancer therapy, and GPCRs may be part of this maybe in immuno-oncology, but not only in immuno-oncology. The EPO pathway, I think, is a very interesting one as well, that where maybe combined therapy, and we've heard a little bit Silvio talk about this. I think this will become more and more important in the future. I would like also to thank Domain Therapeutics, who sponsored this symposium, and as you've seen, maintained it in a purely scientific context. This was not an advertisement, this was a scientific symposium, and I really am appreciative to them for this. It's greatly appreciated. I also would like to thank the participants. We hovered around 160, all the way to 165 participants for the entire morning. It's a relatively long mini symposium, and yet the attendance was there from beginning to end. And so this is remarkable, I think, and it speaks to the quality of the speakers. Also thank them for the participation in the discussion. I know it's somewhat frustrating to have to use the chat and not being able to intervene and exchange. That's why we did the breakout session, to allow that. But with the reality in which we are living in, that makes things much easier. And again, thank you very much for your patience and sending your question. I think it was very dynamic. I don't know for you, but me, I'm leaving this symposium, not quite yet, but soon, with, again, more ideas that I will ever be able to put into action. There was a number of very interesting concepts that were discussed and tools that were presented. And I think the future is bright for the young people who are still in this field or will be entering this field in the next few years. In closing, I really, have seen the names of the people who were attending, many old friends, many people that I didn't know, that I wish I could meet in person. So hopefully very soon we'll be somewhere together and we'll be able to not only see each other by Zoom, but see each other for real and have a beer and reflect on this crazy, 15 months that we just got through. By the time it's over, it will be, more than 18 months. But I'm hopeful that in the fall we'll be able to be somewhere together. With this, I will turn it to Louise so that she can explain to you how to get into the breakout rooms and talk with the speakers. Good afternoon.